that.
Has everybody received
Yeah.
email with the paper?
Yes, I haven't.
Did you look through the papers or paragraph?
Yeah.
Hard to read, hard to understand what the hell they're talking about, right? Well, a little bit, okay. I fully expect that I'm going to be in here on Sunday because people are going to say, I'm having a hard time understanding this.
I feel like if you're just creative, you can read it and make like a magical story about it, about like DNA and stuff.
I can create a magical grade for you.
No, the magical story will help us open up our brain cells to find the solution.
What are you doing this weekend to open your mind?
That would be fun.
It's legal. Not on campus though, right? Okay.
If you're quiet.
So yeah, in the email I sent out, I put my cell phone number. I fully expect you to call. Do not feel like, oh my gosh, I have to call them because I don't understand I'm a lesser being. No, probably every group is probably going to call me. That's just what my Sunday is. Give you a heads up. Sunday night, I'm going to bed at midnight, so don't call me after midnight on Sunday. Monday, I will probably get in here between 6 and 7 in the morning. Again, I would not feel shame for you if you came in at 7 in the morning, 8 in the morning and said, so I'm putting together the presentation, and we're still working on it. We don't understand this. I get it. I understand you guys are busy. So I don't care what you know coming in. It's when you walk out of the class. That's what I care about, right? So today, I want to talk about assays. And I want to develop an assay here. Conceptually, I want to talk about an assay. And then I want to be able to use this. And I want to be able to draw on your data. And then you tell me, what does the data say? What's my conclusion about this? And what does it tell me about the story that I'm trying to put together here? Okay. So, what we've got here, for simplicity's sake, we're going to look at some gene. OG1, OGG1, the OG1 gene. Incredibly, the OG1 gene is all on one exon, the coding part. So if you were to cut this out with restriction enzymes, you put it into a plasmid, you could put that plasmid into cells and you could express that gene just off of that one exon. Okay? Not a big deal. So, let's say I have a gel like this. And I take 100 liver cells, and I break those cells open, and from those 100 liver cells, I purify all of the OG1 messenger RNA. And I run that messenger RNA out of the gel. Got 100 cells, 100 liver cells. And I get that much RNA on my gel. Okay? Let's say I call a company, and I say, I would like you to send me 1 million, I want a tube, and in that tube I want 1 million messenger RNAs of the OG1 RNA. And they send me that tube, let's say that tube, I run that out alongside this, and that is what it looks like. So we know this is my standard for comparison. This is 1 million copies of the OG1 messenger RNA. How much messenger RNA do I have here? Not quantitative, qualitatively. Half a million, you think I got 500,000? Can we all go with that? You're right, it's exactly 500,000. I don't know. Right, we'll say 500,000, okay? Okay, so we know how many messenger RNAs there are in every single liver cell. So what I want to do is I want to say, I want to be able to measure the number of messenger RNAs made in the cell. And my assay is by running it out on a gel and comparing it to a known amount. You guys understand all of that? Okay, so this is the standard type of experiment. This is what I'm going to be running. And now I can start doing some fun things and running them through this assay that I've established, and I can get different reads on it. So, let's take and cut here and here. And that is what I put into my plasmid. And I put this plasmid into cells. And let's put it into, let's say skin cells don't express OG1. So let's put it into a skin cell. And we grow up 100 skin cells. And then I break the cells open and I purify the OG1 messenger RNA from the skin cells. So all of the messenger RNA in these skin cells must have come off of this plasmid, right? Okay. Someone give me a rough idea. How many messenger RNAs are in 100 skin cells? So when I come up with a number? How much do I have if I compare it to these amounts?
1/5.
1/5? Okay, so we've got 1/5 of this or 1/5 of this?
1/5 of this.
1/5 of A or 1/5 of B?
A.
1/5 of A. So we're going to say it's 200,000?
Perfect.
No, I'm going to say 100,000. Let's say 100,000.
Okay.
That'll give me room for the future for what I want to do. Okay. Okay, so this is 100,000. How come I'm not getting the same expression in skin cells in my artificial system as I'm getting over here? Anyone want to make a stab at that?
There must be a regulatory sequence somewhere on the gene.
There's a regulatory sequence on the gene somewhere.
Or like, not on the gene itself, but outside of the area that you cut.
So there's, if I had taken a larger bite of DNA and put that into the plasmid, it would work better. What were you going to say?
Transcription factors?
Yeah, the transcription factors are different. Maybe. Yeah, maybe skin cells don't have the transcription factor needed to express this at the right level. That's another possibility. That's true. But let's explore this. How would you test this then?
You take a larger sequence from.
Yeah. So I keep that original, I keep plasmid A that I made, and now I make plasmid B. I make a brand new plasmid, and I just, instead I use an enzyme that cuts up here. So now, this thing has a larger piece of this, right? Okay, I do that, and I get this. Should we say 200,000 now? Okay, so our plasmid A was this guy. And plasmid B, I added this on, right? So it says somewhere in here is a DNA sequence that allows me to double the expression, right? How could I confirm this with some other experiment?
Is that going to be always the case that you have some kind of like enhancing sequence?
Um, Well, let's get to that in the form of another hypothesis in a little bit. So our hypothesis now is somewhere in here is a sequence that allows me the double expression of the gene that's downstream of it. What are the two ways I can test that? I can mutate it. I can make mutations in here, and I can damage, basically change the nucleotides. So if I change a couple nucleotides here, and my expression level goes back to this, apparently the nucleotides that I affected are the consensus sequence, are the sequence that controls that, yes? Okay, great. So mutate it. What's the other possibility? Move it somewhere else. If I take and cut this sequence and put it in front of some other gene, I should double expression of that gene. It doesn't matter what the gene is, right? And my assay for did I double expression is running the DNA or running the RNA on a gel and seeing the amount of RNA produced, yes? Okay. You asked a question and I said we'd come back to it. Is it always the case that if I increase the size of this? No. Let's make another plasmid. And this time we're going to put a whole bunch of extra on here. We're going to go all the way over here. So plasmid C goes from here all the way to here. Okay? There's plasmid A, plasmid B, plasmid C. So I added a whole bunch more sequence in there, and we're still at about 200,000. Does that answer your question?
I guess I was just curious if like there's always like an enhancing or silencing sequence upstream from whatever you're.
In this case, if it was really close, yeah, but it turns out if you get far away, no.
So it could be in like region C or region B, just.
But yeah, I mean, I could have, yeah. So yeah, this is the question. Is it possible that the results could have been this? Where the thing that doubled expression was located here and not here. Yeah, absolutely. Okay. You guys have any thoughts, comments, or questions about this? Okay, so taking this big, huge piece, and I'm still not even half of what normally happens. What's going on here? Why am I not back? So we have one possible explanation. Transcription factors in skin cells are different. And it's another explanation.
Can they be affected by like the way it's wrapped on like a larger scale?
Yeah, the way the DNA is wrapped on a larger scale. So if it's packaged.
Yeah.
So yeah, it may be that this, if you have a plasmid, the DNA compact differently than a chromosome.
Mm-hmm.
Yeah, that's great.
Multiple genes on different chromosomes that code for the same protein.
Okay, yeah, so there may be 2 copies of this gene in the human genome. Oh, that's great. I hadn't thought of that one.
Is that even real? Like, is that a thing?
Yeah, many human genes appear 3 times in the human genome.
Are they used 3 times? I mean, they're like, yeah.
So let me rephrase that. So, how many of you are trying to clone this gene TMEM106? How many you've got? So we've got people who are doing cloning A, people who are cloning B, people who are cloning C.
Mm-hmm.
It's the same gene of although there are some nucleotide differences in the gene. So the proteins produced probably do different things, but very similar things. That's an example of a gene that appears 3 times in the genome. You get to a gene like myosin, so the motor protein that makes actin do its step, I think that appears 40 times in the human genome. Not all of them have an exact perfect sequence. But if you get to something like maybe histone H1, I'll bet you that gene probably appears 40 or 50 times, and the sequence from time to time to time is almost perfectly the same. So it's common for genes to be repeated numerous times. Yeah.
Can I just ask a question about like our genetics lab? When you said like our genes are like cancer causing, or like they're related to cancer, does that mean like these genes are easily manipulated in the human body to cause cancer, or what does that mean? Like these are cancer causing genes?
So that is the final section of the course, and we do that when we come back from E-Days.
Okay.
You'll learn. I will talk about your gene specifically. Well, not you're doing TMEM. Nobody knows what that does.
Cool.
I think it's a cancer gene. I want it cloned so we can then run some experiments and see. But most of you that you've got bad or back or big or cast-based or something, when we talk about cancer biology and explain how cancer works, you're all going to see your gene in that pathway, in the cancer pathway. Okay. So I'm going to say that all of this stuff here is proximal, is close to the transcript. Let's take this chromosome, really go far, far away on the chromosome. I am 100,000 base pairs, 100,000 kilobases, 100,000 base pairs away from my gene. So what did we learn about promoters? The job of a promoter is to bring RNA polymerase in, so RNA polymerase can measure a set distance that is, it would say like, +1 is here, right? So it makes sense that all of the transcription factors that help RNA polymerase come in and measure +1 are really close, right? Does it make sense to have a transcription factor that binds 100,000 base pairs away? It brings RNA polymerase in, RNA polymerase reaches out, finds +1, makes sense? I am going to say, let's make a plasmid D. Plasmid D, the stuff I clone into the plasmid is so big that the plasmid is like the dinky little part of the plasmid. This plasmid D, I put that in, and I get this. What's the conclusion?
Or something in D that's making it produce more mRNA.
Something in D that gets us up to 500,000. Okay? So let's use that as a hypothesis. So let's state that as a hypothesis. Something in D, something in D what, would you say?
Increases the rate of transcription of this ON1.
I detest it, and you can't talk this time. What's the 2 ways you test it? Mutate it, or move it. Okay? So let's say I make mutations I leave everything the same, but I come in and I make some mutations in this area here. Need another lane. I do that. And I get that. What's the conclusion? It's true. The hypothesis holds. Sequences 100,000 base pairs away from the gene control expression of the gene. Who believes it? 1, 2, 3. 3 gold stars, black stars. The rest. Holy smokes. Okay, what's the other way? Can this really be true? What's the other way of testing? Move it. What if I take this sequence here, the wild type sequence, and I put it on a different chromosome to control some other gene, OG2? If this thing causes things to increase their transcription, if I put that on a totally different chromosome in front of some other gene, 100,000 base pairs in front of some other gene, it should double expression of that gene, right? If you do that, it doubles expression of that other gene. You convinced now? Yeah.
How did you know where on the 100,000 base pairs to mutate that part of the chromosome?
That's an awesome question. How did I know? Out of 100,000, I added 100,000 base pairs on. How do I know to look right here at this end? And the way they did that, it was someone earned a PhD doing it, right? What they did is they cloned plasmid C, or plasmid D, didn't work. Plasmid E, didn't work. Plasmid F, it didn't work. G, H, and they went way up, way up, way up, way up. Way the hell up, they made 200 plasmids, 500 different plasmids, each one taking a larger piece of chromosome with it, until eventually it worked. That's why grad students do the Scott work.
It must have been a good day for them when it finally worked.
Yeah, it takes, oh yeah, I mean, totally, yeah.
And you're saying that's not gene specific, it's like consensus?
Yep.
Okay.
It doesn't matter what gene you put in front of it, it works. Does that sound like fun for your PhD? Probably what happened, probably what really happened, is someone had something going on. They were getting normal expression level, and then the expression level decreased somehow. Like, what the hell happened to my cells? And so they figured out where the mutation occurred, and they're like, this mutation, like 100,000 base pairs away from my gene, is what correlates with my gene going down. Is this really a transcription factor binding site? They restored to wild type, and yeah, expression went back up. That's probably how it happened in reality. Because most things that we discover in biology are like totally by accident. And the purpose of this is for you first to understand again assays. What do I mean when I say assay? Your assay is amount of RNA as measured on the gel. Okay? That's the assay. Okay. So let's talk about promoters here. What were the functions of a promoter that we defined?
Recruit RNA polymerase.
Recruit RNA polymerase. Okay. What else does a promoter do? We talked about this.
That defines which direction the gene goes.
Tells which strand to copy, right? Okay.
How much is copied?
Oh, that's true. How many copies made? And now I'm going to add number 4 in here. Yeah, defines where is plus 1. Right? What we said is if you move the promoter, if the promoter is like this, if you move the promoter over here, plus 1 shifts with it, right? Plus 1 will always be this distance from the promoter, right? So if you shift the promoter over here, then plus 1 will shift to this spot right here, right? That's what we said. It is different. If I took the promoter and spun it around, then plus 1 would be here, and it would go on the other strand, right? Right? We defined that. If you turn the promoter around. Okay. Is this thing part of the promoter? If I take this and I move it to here, do I now shift? So if I take this from 100,000 base pairs to 10,000 base pairs, do I now shift plus 1, 90,000 base pairs that way? We run that, we say that's my hypothesis, this is how I test it. Turns out if I move this to 10,000 base pairs upstream, plus 1 stays the same. Uh-oh. So let me do it this way. Okay, so if that's the direction that it normally is, if I take it and I turn it around so that it's the other way, like that, so if I invert it like I was talking about with this, does it now direct transcription off in that direction instead of that direction? Turns out if I turn it around, this stays plus 1 and we continue to go that way. 1. This thing's not a promoter. I can't, if I tinker with this stuff here, it still works like it's supposed to work. So, we're not going to call this thing a promoter because we've already defined what a promoter is. We're going to call this thing an enhancer. An enhancer can be moved, and plus 1 stays the same. An enhancer can be inverted, and you still stay on the same strand. So it actually says how this thing increases expression is different than how this thing increases expression. Anyone want to guess why this thing has flexibility?
Could it be recruiting helicase?
Could it be recruiting helicase? Yeah, could it recruit a helicase that opens up the DNA so it's not recruiting RNA polymerase? That, I've never even considered thinking of that. That is an amazing hypothesis that I don't think anyone ever tested, and that could be tested. To the best of my knowledge, that's never been tested, and that's a brilliant question. It's not a PhD. And the reason is you can get that done reasonably in a couple months, maybe a year. No way that's a PhD. Maybe you could get a master's out of it. So, the way this thing works is some transcription factor binds here, right? And it brings RNA polymerase in, and RNA polymerase binds to this, measures out its distance, pops down. This thing can't be doing that, right? If you move it different distances, it still works. How can that happen?
Is it messing with the transcription factors to make them work?
Messing with the transcription factors to make them work.
Or I guess
I think you're on the right track.
modifying it in some way, or
Modifying the transcription factors. Where are the transcription factors?
they still be on the promoter.
Yeah, so this is what we call, the definition of this is transcription factor. This guy here is a transcription factor. So can the enhancer somehow be affecting these guys and not at all be involved in recruiting RNA polymerase? Yeah, how's this guy affecting this? I mean, look how far apart they are.
Well, the DNA is all bent up.
Oh yeah, the DNA is this string flap around like that. So it doesn't matter if we're 100,000 base pairs away or we're 10,000 base pairs away. It's a question of how big the loop is. Do you have a small loop or do you have a really big loop? I guess that doesn't work. You have a big loop like this, 100,000 base pairs, or do I move it up to here and it's 10,000 base pairs and it's just a smaller loop of DNA between the promoter and the enhancer, which is the enhancer up here and the promoter down here. It doesn't matter how much DNA is in between because it's just going to loop around and these guys are going to interact like this, right? Yeah. So what the enhancer does, this is just great. So the job of the transcription factor is to bring RNA polymerase in. Helps RNA polymerase find where it needs to go. The job of the enhancer is to help the transcription factor find where it needs to go. Try to describe this. It'd be like someone's pushing a wheelchair, and then the person pushing the wheelchair is actually like in a walker, and has someone helping the person with the walker. There's someone helping the person with the walker push the person in a wheelchair. Does that make sense? This is the wheelchair. This is a person in a walker helping push the person with a wheelchair, but the person with the walker needs help. And so there's a person helping the person with the walker help the person with the wheelchair. It turns out, transcription of a gene, the amount of RNA made off of a gene, there's probably close to 40 proteins that bind to the promoter. And the DNA is looped all over the place, and many things bound to different spots of the DNA all come together to make this huge complex to which RNA polymerase is then recruited and goes off of. Okay, so that's the difference between a promoter and an enhancer. Two additional things to talk about. When somebody found an enhancer, immediately afterwards, someone said, well, gosh, if there's a sequence 100,000 base pairs away that helps turn genes on, I wonder if there's a gene somewhere that has a sequence 100,000 base pairs away that helps turn the gene off, such that if I mutated the sequence, suddenly I'd express a ton off of that gene. And if this is called an enhancer, a thing that increases expression, what would we call something that would decrease expression? We would call that a repressor? Yeah, some people use that term. Silencer. Right. So there are enhancers and silencers. They both work the same way. They're far away, but they loop the DNA around and regulate transcription. Okay. One of the frustrating things that happens in biology, and this is I've actually sat in on lectures. So there are people who study the DNA sequence. And we define the DNA sequence up here as its unit. We can actually define what the consensus sequence is. That thing is called an enhancer. Right? The protein that binds to enhancers are called, can anyone guess? Enhancer binding proteins. The proteins that bind to enhancers. So some people study these DNA sequences, and that's what their labs do. And some people study these proteins, and that's what their labs do. So if I go and I say, okay, so then I was studying this enhancer here, and I made mutations to the enhancer here, and then I was studying this enhancer here, and made mutations here. You understand I'm talking about mutations to this sequence of DNA. if I study the proteins, I don't want to say, it takes too long to say, I study this enhancer binding protein, and I made these mutations to this enhancer binding protein, and then I studied this enhancer binding protein, and I made mutations to the enhancer binding protein. Gosh, that's a real mouthful to say enhancer binding protein. So the people who study this just call them enhancers. So I studied this enhancer and I made mutations to the enhancer, and this is the effect. Studied this enhancer, I got mutations to the amino acid sequence. Did mutations, and I had this effect. There was one time I sat in on a lecture, and it was 30 minutes into the 1 hour lecture. The person was telling me that they were studying enhancers, and I had no idea if they were talking about the DNA sequence or the protein. Sorry. Just prepare you for that. Okay, so, enhancers, silencers, and the language script that people do. That's really what I wanted to cover. Now I want to do the preparation for the microRNA. To shift gears here. Wow, this is not coming off in terms of, that totally sucks. Okay, so when I was in graduate school, we got the nucleotides on a messenger RNA. Messenger RNA. And one way that we turn off translation of this is to bring in, is to make a plasmid which has that messenger RNA backwards. So the transcript that comes off of it is going to be this. So this is antisense. Okay? And the idea is this thing will hybridize like this, such that a ribosome can't come and translate this guy, and we'll never get a protein made off of here. And this antisense technology was real spotty. Sometimes if you expressed an antisense, you completely knocked out expression of the gene. It just worked perfectly. Other times you expressed antisense, it was like this thing couldn't find its partner. And protein expression off of this guy just happened normally. So we had no idea what was going on. But if you wanted to decrease expression of your gene in some cell, you try to antisense, and if it didn't work, you'd say, ah, crap, I wasted all my time doing that, it didn't work. And you just go and try to find some other way to shut down the expression of the gene. So this laboratory, there's a laboratory. When I was applying to graduate school, kind of I applied to a couple dream schools. I applied to schools that I felt like were a good fit for me, like GPA and GRE score. I had a reasonable shot of getting in. And I applied to like a couple safety net schools. Things that I was like, I felt like I was way out of class for these, and I was sure to get into these schools. Does that make sense? So one of those was University of Massachusetts at Amherst. UMass Worcester. University of Massachusetts, the Worcester campus. And I applied, and they were interested in me attending, and one of the researchers called me up, and we kind of did like a little teeny phone interview. And I think I was officially accepted based on that, which is bizarre. Normally they fly you out, and you have an interview, and it's a real big deal. I'm pretty sure the guy I talked to on the phone while I was in graduate school was the laboratory that had this finding, and that guy now has a Nobel Prize. If I had gone to my fallback school, surely his lab would have been the lab I had worked in. And I would have been in his lab when this discovery was made. In my life, I've probably met 20 Nobel Prize winners of medicine and chemistry and such. it's not a big deal. If you should choose to go to graduate school or medical school, these will be your colleagues. These are your everyday people you interact with. You're there. You're at the end of knowledge. You're with these people. Okay? So what they did in this laboratory is, I'm guessing, I don't know, I wasn't there, but I'm guessing this is how it happens. So what they did is they were trying to knock out a gene, and like anyone else, they would say, okay, I've got my normal worm, and I'm measuring how much RNA the normal worm makes, the normal elegans worm. That's how much messenger RNA I make. If I have my normal worm, and I have antisense to my gene, I get much less. Yeah, probably I get less expression of my gene. If I have worm, and instead of antisense, I just express, let's say I express antisense to some other gene. Let's say the gene I'm looking for is OG1. So this is antisense to OG1. Say I do antisense to actin 3. Well, the antisense for actin 3 should have no effect, right? That's control. If I do worm, and I have a plasma that's going to increase more of it, if I'm going to express more of the gene, I would expect to see a larger amount of this, right? It's sense, the sense version of OG1. And I think probably what happened, because I know how things happen in research, I think probably what happened is someone in the lab, so that's a controlled experiment. That's the experiment you run. Somebody in the lab probably went super control and said, I'm going to have my worm, and I'm going to add antisense, and I'm going to add sense for OG1. So you'd say the antisense and the sense should knock out the normal copy in the worm. I would do this, right? I'd get the normal expression level in the worm, right? And what they got was that. Someone tell me why. What's the conclusion?
All the sense and antisense combine to make some sort of, to destroy the normal gene, I guess.
Having sense and antisense both somehow allows the normal gene to be identified and destroyed better. Yeah? The normal copy of the gene is destroyed more efficiently when you have sense and antisense in them both. What kind of mechanism could be involved in this? Sense of using your brain, what are the sense and the antisense doing? You put them in the cell. They're what?
Belonging to each other?
Yeah, they're bonding to each other. So you would think that it's the double-stranded nature created by the sense and the antisense. Somehow giving a double-stranded version of a gene causes that gene to get shut down. So this is probably the experiment they ran in their lab. Nobody in their right mind would run this control, this link. No sensible researcher would waste their time running an additional tube with all of this crap in it and putting it together and bothering with it. But somebody in the lab did, and they got the results. And if it were me, I would have said, oh, you know, that's weird. And I probably wouldn't have paid any attention to it. I said, you know, so I ran this experiment, my antisense seems to work a little bit, so we're going to run with it, with what I was originally planning. Somebody in that lab said, what the hell? That's incredibly valuable. if we can figure this out, maybe we could maybe we get antisense to work for everybody. And so the paper that you are now going to be presenting on Monday and Wednesday is them elaborating on this and showing to the world there is an active cell process that definitely makes this better than this. And we understand it in excruciating detail now. It's incredibly effective. And it took them 6 years from publishing the first indication that this thing existed to winning the Nobel Prize. Okay. Yeah.
How do you know when you do something like that, that you just didn't screw up the experiment?
You only need to have controls. So, yeah, so
Just
the question is, was there a pipetting error here? Did they forget to load their sample? Hey, that's why it looks like there's nothing there. There's no sample in there, right? So, I mean, that's when you say, man, I don't understand these results. And you go back and you run it again, and again, and again. And you say, okay, well, you know what I'm going to do is I'm going to take this same goop and run it and look for some other protein. Well, that protein looks normal. And so it's not like I missed this, or the protein in here got degraded or something like that. So you just run control, control, control. And probably what happened is someone did this, and my guess is they presented at a lab meeting. They said, okay, so my antisense works, so off I go on continuing on my project. And someone in the lab, probably the PI in the lab, said, wait a minute. That's more important than what you're getting your PhD on. Let's look at that. And probably the student found it was like, look, I'm like a year away from my PhD. I don't want to start something new. Let me just finish this. And so he's like, okay, I'll take the next grad student who comes in. Is just going to come in and get a Nobel Prize project. See ya. That's probably how it all plays out. Okay, that's what I've got. I look forward to your presentations on Monday. I've got a challenge for you guys. In my botany class, I used a meme in my